Hello everyone, my name is Tyne Lee and thanks for visiting my virtual poster. I will walk through our multitask matrix factorization method to detect higher order structural changes in 3D genome organization data. A 3D genome organization refers to how the DNA is folded in a systematic way inside the cell's nucleus. Distant regions driving gene regulation, like enhancers and promoters, can be brought to close proximity in 3D space. And we can study 3D genome organization with hESC contact count matrices. These measure the level of interactions among genomic regions, including enhancers and promoters. It generates a symmetric matrix like the one you see on the right, whose entry represents how often the region corresponding to the row interacts with the region corresponding to the column. An analysis of hESC data has revealed higher order structural or organizational units on multiple scales. We have compartments, topologically associating domains, or TADs, which are these groups of highly interacting neighboring regions, and at the most granular scale, chromatin loops. We're in particular interested in changes or dynamics at the TAD scale. And what motivates this interest involves how TAD level changes can lead to diseases, including cancer. So here on the left, we see an enhancer in red insulated from accessing a gene in a different TAD. And disruption to this TAD boundary leads to this ectopic enhancer-promoter interaction, leading to the activation of an oncogene. TAD scale changes are also observed throughout the differentiation process. Shown here is the hESC data from mouse neural development time course, from mouse embryonic stem cells to cortical neuron, and we see corresponding gene expression changes in the vicinity. TAD scale changes are also have implications for evolution. So a key challenge in regulatory genomics is to gain a genome-wide understanding of how structural dynamics can have downstream consequences from genome-wide data sets like hESC. And we need an effective way to narrow down and focus on regions that are specifically involved in TAD scale changes. So we distill this goal down to a computational task where we have multiple input hESC data sets from a related biological condition or from a biological process. And we want to provide biologists with an interpretable output that annotates each genomic region with some context-specific boundariness. So here in an example output bed file, each row represents a genomic region, and it has some annotation about whether a shared or a context-specific boundary is found within that region. And to this end, we've developed a pipeline that involves a multitask matrix factorization framework with some downstream significant testing and annotation, which I will briefly walk through now. In TGIF, or tree-guided integrated factorization, each task represents a biological condition. TGIF performs a joint factorization of multiple input matrices to yield these lower dimensional representations of the genomic regions, or these are called factors, and these factors obey the structure of the user-specified task tree. And this task tree can encode the relationship among the tasks, for instance, cancer subtypes, if these are hESC data sets from cancer cells, or cell lineage, if these are from different cell types during differentiation. Once we have these factors, we calculate a boundary score for each of the regions. The boundary score measures how much the interaction pattern of a given region I in this lower dimensional space captured by the factor U changes from its immediate upstream neighbor I-1. This essentially, the boundary score is capturing this cosine distance between two neighboring regions. This boundary score should reflect changes in the interaction patterns back in the high-dimensional interaction matrix, in the high-dimensional hESC interaction matrix. And we can then perform significant tests to determine essentially meaningful boundaries or significant boundaries. And finally, once we have identified the significant boundaries, we can then annotate each region for their context-specific boundariness or their persistence in the domain boundariness throughout these different biological contexts. And then we have our final output bed file where each row corresponds to a genomic region, and we have some annotation about whether a shared or context-specific boundary is found within that region. We benchmarked TGIF to two existing approaches for identifying differential TAD boundaries using simulated hESC data sets. We see that TGIF yield in pink, it yields better precision and overall higher F-score in both task-specific as well as common boundaries over different degrees of differences between the two tasks or two input data sets that are being compared. We next applied TGIF to real hESC data collected from 6 different time points during cardiomyocyte differentiation. Zhang et al., which generated the original data set, they had found that transcriptionally active HERV-H retrotransposon sites, they actually demarcate TAD boundaries specifically in human pluripotent stem cells. And here we actually visualize one such HERV-H site that has a TGIF boundary that is only specific in human embryonic stem cells. Finally, we've started to analyze disease-associated GWAS SNPs and their relationship with a differential or persistent boundaries that are identified by TGIF. So among about 4,800 GWAS SNPs that are associated with cardiovascular disease in the GWAS catalog, about 10% were found in a boundary in at least one of the time points during the cardiomyocyte differentiation. Here as an example, again, we show 2 GWAS SNPs and their associated genes that are falling on a shared or persistent TGIF boundary. And we plan to further analyze these SNPs and demonstrate how TGIF can be used as a hypothesis generation tool in understanding variation. I'd like to thank my advisor Sushmita and the members of the Roy Lab who were able to do our research thanks to the funding from the NIH and the CIBM training program, as well as the computing infrastructure maintained by the Center for High Throughput Computing. Thank you for listening.